Hi class, I am going to give you a review for your second lecture exam. Your lecture exam is on Monday. Monday we also have discussion. So what I want to do on Monday is go over muscles of the head, eye, and neck, and finish the muscles of the thigh. And then after that, you will take your exam. So I will not be using more than 2 hours of our 4-hour class time if you add our lab and discussion. And that will still give you 2 hours to take your exam. So that's what I'm planning for Monday. I would review, I'm going through the going through the lecture exam now. I would review your homework packet for sure. And there are 100 possible points on this exam. It will be graded out of 95. Yes, Terry, there will be a word spell. I have to give you one after last exam where I didn't come through for you. Anyway, I would review your structures of your heart, very comparable to what you saw in your homework packet and also on the quiz for the heart. I would review your tunics for your blood vessels. What are they made of? What kind of tissue are they made of? Which layer is made up of smooth muscle, for example? And what does it do? How important is that middle layer? What's the difference between an artery and a vein? They have characteristics that are unique to both. So please review what your arteries look like, muscular versus elastic, and what veins look like. For example, veins have valves, but arteries don't. Please review what you find in plasma, meaning suspended, dissolved in plasma. What do we find in plasma? When it comes to the heart cardiac cycle that I spent one of our discussion times on going through it and telling you that it was arbitrary where I was starting that cardiac cycle. And I started at diastasis when all 4 chambers were relaxed. I would know when your valves are open or closed during all of those phases of the cardiac cycle. What is the definition of anemia? I really want you to know that. And what is a hemocytoblast cell? It is a type of stem cell, and it is determined, but it is not fully differentiated. It replicates and divides and gives off daughter cells that will in turn differentiate. But the hemocytoblast itself, it's determined again, but not differentiated. It can't generate anything beyond blood cells. So that's what we mean by determined. But it is certainly not mature and differentiated in any way. Please review what it means when I tell you about exchange vessels or capacitance vessels and resistance vessels. I did that vessel lecture with you all live, and it is posted on YouTube now. I don't know if you've noticed that, but it is. Where does blood cell synthesis occur? It changes through development. Where is it in the embryo? Where is it in the fetus? Where is it in the adult? And know what hemoglobin is. Moving on to lymphatic vessels. Again, very similar to the quiz, very similar to your homework packet. Know your lymphatic vessels. Which ones are the smallest? Which ones are the biggest? What is the infection called where we get roundworms living in the lymphatic vessels and it leads to edema? Please be able to match your formed elements, meaning red blood cells, white blood cells, platelets, to their descriptions. Again, this was very similar in your quiz. It was very similar in your homework packet as well. So, for example, never let monkeys eat bananas. What does that mean? N stands for neutrophil. It's the most abundant granulocyte. And we remember granulocytes because they're filled with granules. So anything that ends in a fill is a granulocyte. Agranulocytes, meaning they don't have granules, would be your B and T cells and platelets, right? Platelets, though, are not white blood cells. How many molecules of oxygen can you transport on hemoglobin? Again, know your anatomy of your hemoglobin. And what is the hormone called to stimulate erythropoiesis, and where does that come from? There are two sources of this hormone, and there's one big source. So please know the source of the hormone that would stimulate erythropoiesis. How long do red blood cells live? What is a normal hematocrit? What is a normal hemoglobin grams percent? I would know the statistics on red blood cells that I gave you in lecture. When I was lecturing on blood vessels, I told you that there were 3 different kinds of capillaries. Which type of capillary is the most abundant, but least permeable out of the 3? And which one is least abundant, but the most permeable out of the 3? And where would you find these vessels? Okay, so again, lymphatic vessels, what is a lacteal? You should know what a lacteal is. How would you describe the thoracic duct or right lymphatic duct? What do those things drain from? Areas of the body do they drain from? What are tonsils? What is malt? Also referred to as galt, M-A-L-T, galt, balt. What is the importance of your thymus? And when we're talking about lymph nodes, please know that there are afferent vessels coming to the lymph node and efferent vessels leaving away from the lymph node. And what is the role of the spleen? There's red pulp and white pulp. What's the difference? What's going on in those 2 areas of the spleen? Moving on to respiration. You should know the muscles for quiet inspiration, which muscles need to contract. And then for quiet exhalation, what muscles need to contract? None. We only have to relax the muscles of inspiration when we're doing quiet exhaling. And then how is this different for forced inspiration and forced exhalation? What's the problem if a baby is born prematurely with their lungs? What's missing? What are the parts of the brain that regulate breathing rate and depth? Please review your chemoreceptors and baroreceptors. First of all, chemoreceptors and baroreceptors are found in the same place. They are in the wall of the aortic arch and the carotid sinus. The carotid sinus is a blood vessel. The common carotid comes up and then it splits into the external and internal carotid artery. And where that split happens, it's called the carotid sinus. Baroreceptors detect stretch. That's what they do. When the heart ejects more blood, then there would be more stretch of the aorta and the carotid sinus. Remember, the aorta is an elastic artery, so it's really good at stretching, but also recoil. So baroreceptors are going to detect stretch. And if your blood pressure goes up, then it's the medulla oblongata that's going to send action potentials down to your heart to reduce heart rate and force of contraction. Chemoreceptors are found in the same place, and they're monitoring the levels of oxygen. So if your levels of oxygen go down, again, these chemoreceptors are going to send information to the medulla oblongata. And this in turn is going to result in an increase in breathing rate and depth. You need to oxygenate more. Please review the parts of your respiratory system. You should be able to trace a molecule going through your nose all the way down to the alveolus and into the bloodstream. Now, there was some concern because on your study guide it says to know the blood vessels from your hit list, your lab hit list. Well, we're not to lab unit 3 yet where that hit list is available to you. So that clearly is not a fair thing for me to test you on when you haven't learned about those blood vessels. But what is fair, the blood vessels I can go after would be the coronary blood vessels, like the anterior interventricular artery, the left and right coronary artery. That definitely was in the lecture. It was also in your homework packet. So knowing the blood vessels that serve the heart wall is important. You should also be able to describe the blood vessels in general as you go through your body, meaning elastic artery to muscular artery to arterial to capillary to venule to vein to vena cava. That you should be able to tell me about. The pulmonary circuit, same thing. Pulmonary artery or pulmonary trunk leading to the left and right pulmonary arteries, leading to arterioles, leading to capillaries, which is where gas exchange is going to occur between the capillary and the alveolus, back to pulmonary venules, pulmonary veins. They're paired. There are 2 right and 2 left, and those are returning oxygen-rich blood to the left atrium. So those types of blood vessels are fair game. Why do we need cartilaginous C-rings in our trachea? What are they there for? Again, what's wrong with a premature infant's lungs? What cells do we find in the alveolus? There are type 1, there are type 2, there are macrophages. They all have an important role. What's the part of our respiratory system in the larynx that keeps food out of our trachea? So I would review the cartilages that you find in the larynx, know which ones are elastic versus a mixture of elastic and hyaline. Air flows into our body because we cause volume changes, which in turn lead to pressure differences, and then there's airflow. So please review what happens to the size of your thoracic cavity when your diaphragm is contracting, and which way is air flowing? Is it flowing into or out of your lungs? Each lung is surrounded by pleurae. What are pleurae? These are some of our membranes that we studied from lecture unit 1. What is the condition called if there is air in the pleural cavity. And what would that lead to? Why is air in the pleural cavity not good? And then in your respiratory lecture, I talked about the conducting zone versus respiratory zone. Please know the structures that are in the conducting zone versus respiratory zone. And again, review your red blood cell and white blood cell descriptions. And you will have 10 points where you're going to have to do some sort of diagram flowchart. Now, when you do this part of the exam, what you're going to do is write it on a piece of paper, right? You're allowed to use paper. When you take your exam, you hold up your paper and you show me the front and back. You are going to be diagramming blood flow through the heart, to the heart, through the heart, into systemic or pulmonary circuits. Again, this is in your homework packet. It was demonstrated to you. And you will also need to tell me about fetal blood flow. So again, you're going to hold up this piece of paper front and back, and then be ready to follow my instructions to diagram this blood flow and also fetal blood flow. So each question is going to be worth 10 points. Okay, class. Well, I think that's it for our lecture 2 review.